This is iGEM, the International Genetically Engineered Machine Competition. Every year, thousands of students across the globe build synthetic biology projects to tackle real-world problems, from disease to climate change to food and nutrition. At Cornell University, we're one of those teams. We've done cool projects on oncology and diagnostics in the past, and this year, we're tackling something out of this world. Our 2025 project is called Prosper, a system to make Martian soil safe for plants by filtering out ions that are toxic in large quantities. We asked 3 of our sub-teams what they do to build Prosper, each from their own level of expertise. First, we have Wet Lab, students who engineer the microbes. Then, we have product development, students who design the delivery system. Finally, we have policy and practices, students who ensure our solution is ethical, safe, and grounded in reality.
Can you talk a little bit about what you do as Wet Lab?
So there are multiple sub-teams within the project team iGEM, and Wet Lab primarily focuses on the biological and genetic components of the team. So we deal with bacteria using different synthetic biology tools like PCR, transformation, Gibson assembly, and to assemble different genetic circuits. And we also deal a lot with computational modeling to ensure that the work that we do in the lab can also be applied in a larger scale.
So what's your plan for cleaning Martian soil?
So what our plan is currently Martian soil is very inhabitable for plant growth, and what we're trying to do is we're planning on running water through the Martian soil, and there are a lot of perchlorate ions in this soil, and we're trying to remove this perchlorate from the Martian soil by genetically engineering E. coli to express perchlorate reducing genes so that the perchlorate in the Martian soil is reduced to chloride ions, which we're having a co-culture to have a different type of bacteria to remove these chloride ions from the solution so that Martian soil can be used for plant growth.
How do you know that this will work in a real environment?
Usually when we're trying to solve a real world problem, we see if it works on a smaller scale first and try to apply it to a larger scale. So as of now, we're trying to see if this works on a more smaller scale with a few bacteria, a little bit of soil at a time. And if this successfully works, we then have to think about how we would modify different parts to make this more applicable to a larger scheme of things.
What's something that went completely wrong at first?
At first, we did have a little trouble trying to genetically engineer the E. coli to take up the plasmid with the perchlorate reducing genes. However, we are trying different troubleshooting methods like using calcium chloride to ensure competent cells, and hopefully that works well.
How would you Oh wait, can I spring a question onto you?
Yes, you can spring a question onto me.
Can you talk a little bit about what you do as product development?
So in product development, we basically design a bioreactor system to sustain the bacterial co-culture that our wet lab sub-team designs.
How would you design a system to help Martian soil become plant ready?
So for it to become plant ready, we first have to remove certain substances from the soil, one of which is perchlorate, right? So we currently are working closely with Wet Lab to design, like I said earlier, a bioreactor system that can sustain a bacterial co-culture to process those perchlorate ions and remove them. Now this sounds very fancy, but really all a bacterial co-culture is are two species whose inputs and outputs are linked. So the input of one bacterium, the input to one of the bacterium's metabolic cycle, is the output of the other, and vice versa. And so our bioreactor system's sole purpose is to develop and maintain some form of hardware that can keep those bacteria living and functioning normally.
What were different iterations of your design, and what do you think you guys had to consider the most about feasibility?
So product development is more engineering heavy sub-team. So as you can imagine, we iterate through many designs. We initially had a liquid culture design, which again, that might sound fancy, but all it is the bacteria in the bioreactor, right? In this chamber, the bacteria are in the liquid, right? And there was a number of issues with keeping the bacteria contained. And so we designed a new system with a carrier. And so this carrier, we will still have a liquid portion of it, but this carrier will essentially be some solid device made of plastic or similar materials, on which biofilm will be grown initially and then put into like a treatment chamber. What a biofilm is essentially a layer of cells. So we will grow that layer of cells on the biofilm and then put it into the reactor. And that way, we are, at least at this point, we believe that most of the bacteria will remain on the carrier, and that way we won't have to worry too much about bacteria leaving and exiting the system and modulating, the various modulating the intake of media and liquid to maintain this bacteria in that reactor. So the carrier really solves that. So we ultimately decided to go with not like a 3D printed lattice, but a piece of glass wool, which in the literature has been shown to be very robust, which has been shown to promote a robust biofilm growth and really ensure that not a lot of the bacteria are leaving. They're staying on that piece of glass wool, and that the biofilm is growing. It is not just staying, but growing quickly, which is another thing we had to worry about is the effectiveness of the growth.
And lastly, if the system had to survive a Martian dust storm, what would break first?
So the idea is for this system to be housed in an enclosure. So the first damage that would occur would be to the external walls of this enclosure, right? But if the bioreactor were exposed, for lack of a better term, a lot of bad things would happen, right? You could have the pumps failing, you could have we have made it to be a continuous system that is not really open to the outside world. But I suppose, as things break down, you could get dust that gets into there. And a lot of the stuff that we've worked with PNP, for example, a very integral member of our project team, has been to evaluate the effects of, leaving these bacteria just on the Martian surface, right? And this is not a very good idea. So making it, safe from dust storms is very important. But I suppose an enclosure, housing the bioreactor system would be very good for that.
Your sub-team doesn't build the bacteria or the bioreactor, so what do you do in this project?
So our sub-team, first of all, is called Policy and Practices, and you may be wondering what that is. So I'll try to clarify that by explaining. So Policy and Practices, we look into two different things that we can do. First one is outreach, and second one is education. Outreach involves a lot of different activities, including going to senior living centers so that we can talk to people who are maybe more knowledgeable than us, or people who might have questions, and we can discuss that together and maybe form some kind of ideas that we can apply to our project, and further think about things that'll appear in the future where more people might be asking questions about that. And then education, we talk to a big range of people, including children who are at a very young age but are very curious about science in general, and we want them to get to know more about synthetic biology early on so that it sparks their curiosity, and also so that they can explore that field without having a lot of confusion or without being scared.
So what makes policy and practices different from just doing background research?
Policy and practices, we do background research, but we also do a lot of other things as mentioned before. We reach out to a lot of people, but we also do a lot of things regarding writing policies that are not available right now to the population, because some things or some points may be overlooked when we look into policy. And this is because not a lot of technology is developed and we're not looking straight into the future. So what we do is we look into what we can produce, like bioreactors, and with that what we can do in places such as Mars, like outer space. And then we write policy so that we can provide people straightforward guidelines once we reach that point when people can live on Mars.
And so what's one of the big risks that your team had to think about that other sub-teams might have missed?
One thing that we were really pondering upon was the big why SynBio question, and this question is really important to all the sub-teams, but it may have been, again, overlooked because it's such a basic question. But since it's so fundamental, we had to look into it deeper as Policy and Practices team. And what we did was we collaborated with the business sub-team, and then we dissected each part of why SynBio by looking into things like what the terrain is like on Mars, what the climate is like, and also we looked into things like the ethics of going to Mars or going outer space and using microbes that are currently on Earth but maybe not on Mars or anywhere else in space. So there are risks and dangers to that, and we tried to do a lot of research by looking up papers and also interviewing stakeholders and professionals in that field, like experts such as professors at Cornell and outside of Cornell, so that we can gain a lot of opinions on experienced people.
Okay, so why SynBio? Why use synthetic biology to solve this issue?
So SynBio is being used in our project because there are a lot of problems and extreme challenges on Mars right now. Like there's no water on Mars that we can readily use when we're trying to use our bioreactor there, and it's also really cold on some spots on Mars because there's barely any sunlight on it. And perchlorate-rich soil, obviously that's the biggest problem that we're trying to tackle here. So that's why we're using SynBio. We're using SynBio because it's an adaptive solution and it's sustainable once we do it correctly. We're going to engineer microbes like E. coli and Cyanococcus to detoxify the soil full of salinity and perchlorates. And by using that, we will be able to effectively reduce salinity. And unlike hydroponics or other types of filtration, this way of doing soil remediation is less intensive in terms of putting in a lot of effort to maintain the bioreactor, and also it's less resource intensive, meaning that once the microbes are set up, we have to take care of less things compared to when we use other methods like hydroponics or other filtration methods.
What's the biggest misunderstanding people may have about your sub-team's role?
So when I tell people that I'm a part of Wet Lab, they usually think that I spend the majority of my time in an actual lab doing lab work. However, we do a lot more things outside the lab. For example, the plasmid, we have to design the plasmid first with all the necessary components. This is all done on our computers. Another thing, we do a lot of literature review to ensure that what we do is actually correct. We spend a lot of time planning out what we do in the lab before we go in. Usually when we start doing things without planning things out in a very step-by-step manner, something goes wrong. So we ensure that we understand everything that we're going to do in the lab that day. So there's a lot of planning and thinking beforehand that goes into wet lab work.
What's the biggest misunderstanding people have about your sub-team's role?
That we're a bunch of simply hardware people. But as you can see, we have worked quite a bit with the biofilm. We work very closely with Wet Lab, sometimes doing things that Wet Lab does. We focus on the more business side of things as well with the business team. So we're very interdisciplinary, and we have to focus not on just the technical aspect, but the selling aspect of it as well. Because ultimately, if our product doesn't sell, then it's no different from just a random research project. But yeah, product development is not just a bunch of hardware people. It's a lot of other things, a lot of other considerations.
What's the biggest misunderstanding people have about your sub-team's role?
I think that our sub-team is known for doing a lot of research online, maybe on the computer all the time, but that's not what we do all the time. We might be doing that sometimes, but a lot of the times we are out there in our community reaching out to people who want to learn more about synthetic biology and want to learn more about our project. And because these people are curious, but they're wandering out there, not knowing what we're doing, we want to go to places where we can meet those people. For example, we went to the Science Center where we could meet a lot of families with young children who were curious about space and biology. So we were able to conduct a lot of activities, and we were able to be really friendly, and we were also conscious of how they didn't have a lot of background knowledge, so we tried to simplify a lot by explaining things in the ways children would understand. And we would also introduce our children's book to those kids so that they can enjoy learning about synthetic biology. And other than education and outreach, we do a lot of interviews on campus and online as well through Zoom, because we want to talk to as many stakeholders as possible and understand different perspectives about on things that we do research on. So we would do interviews with professors in plant sciences or crop and soil management on our campus, but also we would reach out to people involved in NASA and other space companies. So it's really insightful to listen to what they talk about, and we apply that to what we document.
And do you think your solution could work without others?
Definitely not. So all the sub-teams are very important in our current goal. For example, we work a lot with PD. PD works with biofilms and ensuring the whole contraption works. So Wet Lab, we deal more with ensuring that the bacteria part of the project works out, that the bacteria has perchlorate reducing genes and the bacteria can survive together. whereas PD, they work more with creating a habitat for these bacteria and creating a system in which we can filter the soil properly. Other sub-teams, PMP, they ensure that that we follow safe protocols in our project, that there aren't any ethical concerns with what we do. Also talking to other experts in the field and help us troubleshoot any problems that we have. Business and wiki, those sub-team, they also play a big role in that they help organize the data we have. Like wiki organizes the data we have and organizes in a way that other people can more easily understand through our website page. Business, they ensure that everything on the money side works out, and they've been very helpful in obtaining all the bacteria, like the primers, everything we need in wet lab. So it's all very important.
And lastly, could your solution work without the other sub-teams?
So what I'll say is this, right? iGEM is created from many, or iGEM and our project, Cornell iGEM, is sustained by the collaboration of multiple different people. As much as Wet Lab or Product Development may think like they're the central sub-team, it's really not the case, because our PNP people, our business people, wiki, all of these other sub-teams are also very integral in presenting the product, in outreach, and they also emphasize the more soft side of the engineering team, and that is absolutely critical, and that cannot be understated. So I think that our project is very interdisciplinary and requires the work of multiple people, multiple hands. So yes, definitely, we would not be able to do what we do without the generous time commitment of these other sub-teams.
And could your solution work without other sub-teams?
Without other sub-teams, I believe that it's impossible to do all this, because we all collaborate and put our work together so that we can have a one polished product at the very end of our season. So with the knowledge that we gain from other people that we talk to, and from the research that we do on our own, and from our discussions that we do during our meetings with other sub-teams, we contribute to the entire team by letting people in wet lab, product development, business, and wiki know about what we know, so that they can apply that to what they do, such as producing the bioreactor and doing experiments on E. coli and Cyanococcus.